Hi, my name is Steven Robbins, and I'm a researcher at the Australian Centre for Ecogenomics in Brisbane, Australia. And today I'm going to tell you about our work in constructing the Great Barrier Reef Microbial Genomes Database, or GBR-MGD for short. The idea of the GBR-MGD is to create a comprehensive database of microbial genomes for the Great Barrier Reef. The initiative is a joint partnership between the Australian government's Integrated Marine Observing System, or IMOS, the Australian Institute for Marine Science, or AIMS, and the University of Queensland, where I'm at. So why do we want to create a database of reef microorganisms? Firstly, because Australian marine systems are quite understudied. So this is a graphic from the Ocean Microbiome Database, which contains about 35,000 microbial genomes from the world's oceans. And as you can see, sequencing has mainly occurred around like the Americas, Europe, Africa, but only a few samples could really be considered to be from Australia rather than the open ocean, and none are from coral reefs. So reefs are really understudied, as are Australian oceans. The other reason we wanted to do this is because there's a lot of interest here in what we call microbial bioindicators. And the idea is that microbial communities shift predictably in response to environmental change, things like marine heat waves, nutrient runoff from farms, or any other disturbance that you can think of. So if we can find microbes that reliably indicate disturbance, we can use them as monitoring tools. But to find them, we need a database first. So to make the database, we linked up with AIMS's long-term monitoring program, or LTMP, to collect reef seawater from across the Great Barrier Reef. And the LTMP is actually like the perfect partner to work with because they've been collecting time series data across the Great Barrier Reef for decades, things like nutrients, temperature, coral cover, things like that for ages. So our original goal was to generate 10,000 microbial genomes that could serve as a baseline for future change. So in the same way that we survey coral reefs to know how the reef is changing over time, we want to be able to do this for microbes as well. And then we kind of had this stretch goal where we wanted to extend the GBR-MGD to include viruses, plasmids, and eukaryotes. So I have to take like a detour here, a bit of a side quest, because I want to hammer home this point. The goal of getting 10,000 microbial genomes from seawater was a huge problem because we knew that microbes don't sequence well microbes from marine systems. So for over a decade, people in metagenomics have been discussing this weird phenomenon where if you survey marine microorganisms with microscopy or marker genes, you can see that by far the most abundant things are microbes like Pelagibacter, Prochlorococcus, SAR86, and a few others. But when you try to sequence these things using metagenomics, you get no genomes, or very, very few. And I've highlighted a few papers that have pointed this out. You can see in this kind of seminal landmark paper from Pachiadaki, they point out that no good Pelagibacter genomes could be recovered from this huge global data set of marine microbes. And Pelagibacter are one of the dominant lineages across the entire ocean. So not getting any good Pelagibacter genomes out of tens of thousands of sequenced genomes from hundreds of samples means something's very systematically wrong. I also did this informal survey on Twitter where I asked my colleagues whether they'd seen this same thing happen in other taxa, and they do. So for all the things that you would expect, the dominant things like Prochlorococcus, SAR86, and Pelagibacter. And I'm calling these ghost taxa. So they're kind of like the things that you know should be there, but you can't see them. And the theory of why this happens is something like maybe these microbes show really high number of strains that are present in each sample, which is to say in each droplet of seawater, there would be 1,000 different versions of the same genome, and that would really confuse short-read genome assembly software. And so we kind of took a shot when we started this and said, look, if this is true, because it's never been tested, it's just kind of a hypothesis, is that this is a problem with short reads. And we know that all of these previous large marine genome sequencing efforts have used Illumina short reads exclusively, so almost exclusively. So if our problem is strain variation and short reads don't play nicely with that, what happens if we use really long reads? So nanopore reads can reach up to, I think the record is 400 million base pairs at this point, and that's a record. It's not the like every read you get. But if we have reads that nearly span whole microbial genomes, that means that assemblers don't have to make a lot of decisions and assembly becomes kind of trivial. So we chose 27 samples from across the Great Barrier Reef to send for nanopore sequencing. And one thing I have to point out is that our data is a bit old at this point because nanopore moves so fast. It's R9 chemistry. So when we refer to our nanopore assemblies, we're talking about hybrid assemblies of nanopore and Illumina. But we also have a follow-up study using nanopore's latest R10 chemistry and the newest super accuracy base calling models, newest assemblers. And I'm pretty sure the results are going to be the same, and I expect them to be much better. So getting to the results, do we see an improvement using nanopore? So what we did is we wanted to test this explicitly. We grabbed 8 samples and we generated what we call metagenome-assembled genomes, or MAGs, from, on the one hand, 30 gigabase pairs of Illumina-only data, and then we did another hybrid assembly with 15 gigabase pairs of Illumina data and 15 gigabase pairs of nanopore data that combined make 30 gigabase pairs. So what did the nanopore assemblies buy us? Well, we saw 2 times the number of MAGs, 3 times the number of high-quality MAGs, and the N50 of those MAGs went up by about 10 times. And so that was great. But the real question was, did this increase in MAG recovery include our ghost taxa? That's what we really wanted to achieve. And the answer is yes. Like, really, it really worked. There are several marine prokaryote groups, it turns out, that you simply can't recover with Illumina that you can get with nanopore. And we even found a few new ones that hadn't been reported in the literature. So what this graph is showing is the number of MAGs per sample that we can recover from each of these groups of ghost taxa. So these are all ghost taxa. And it's worth pointing out these aren't individual species, they're whole families or even whole phyla. And the difference is pretty stark. So for example, the top row here are all different groups of alpha proteobacteria, including Pelagibacter. And in all of these cases, you really can't recover their genomes using Illumina, but you can get as many as 14 per sample using nanopore data. We also found that you can't recover MAGs from the most abundant microorganism on the reef, a specific species of Parasynectococcus, unless you use nanopore. You can get a few Parasynectococcus, but you can't get the most abundant one. If you want to use, so if our goal was to make this super useful database of marine microorganisms, that's pretty important. So I think, the list goes on of the different things I could say about this slide, but I'll have to leave the details here. To sum up, though, I think that the crux is this. So yes, nanopore sequencing does buy you high quality MAGs and higher quality than Illumina, but the real value proposition is that you're missing almost everything important if you don't use it. So for us, that was huge, and it was kind of a no-brainer. So we also wanted to revisit the question of why this happens. And in looking at all of our ghost taxa, the main thing that sticks out immediately, or did to me, is that almost all of them have GC contents lower than 40%. And so we mapped the Illumina reads to our MAGs, and we highlighted the ghost taxa, and that's what I'm showing here on the right plot. And what we found is Illumina has a strong GC bias below about 40%, and so it's really biasing against our ghost taxa. So the Marinosomatota, which is a whole phyla, are as much as 25 times underrepresented in the Illumina data compared to the nanopore reads. And we also calculated strain diversity metrics for our MAGs and then plotted those values against GC. And we found that above about a value of 0.01 strain diversity, which is a kind of a fancy metric for SNP variation. So above 0.01 strain diversity and below about 40% GC, Illumina kind of enters this black hole where no genomes can be recovered. And we've highlighted this here in the left 2 plots. Which the one in yellow is Illumina, where you can see kind of the black hole. The one in right is nanopore and highlights where all of our ghost taxa sit. And all of our ghost taxa fall inside this range. So what we're seeing in the end is a platform-specific incompatibility with our samples. And we were really kind of happy to be able to answer the question of why this happens. So in the end, we were able to recover more than 5,000 bacterial and archaeal genomes from the Great Barrier Reef. About 1,500 of those were near complete, and about 350 were completely circular, including Pelagibacter, some of them. So we went from not being able to recover Pelagibacter to being able to recover complete Pelagibacter. And interestingly, when we compare our MAGs to the 35,000 microbial genomes from the Global Ocean Microbiome Database, which is a collection of all previous studies, we find that 2/3 of the GBR-MGD species are new, which is great. And so what can we do with this database? Our main goal was to identify microbial indicators of ecological shifts or changes. So as a test case, what we wanted to do is see if we could take a sample from a random reef and predict whether that reef was open or closed to fishing under the Great Barrier Reef Marine Park Authority's management plan. That's always hard to get through. And we can with about 72% accuracy. So if you handed our student, Marco Turzin, a random sample of coral reef seawater, he could tell you whether that reef was open or closed to fishing without knowing what the reef was. And the interesting thing about this finding is that all 120 of the taxa that ended up being good bioindicators of fisheries management strategy were ghost taxa. And that's important because it means that we could only have achieved this result using nanopore reads. The next thing we wanted to achieve as kind of our stretch goal with this database was to extend the recovery to viruses and eukaryotes. And I've left our viral findings out for another time. But I wanted to include the fact that we were able to recover telomere-to-telomere eukaryote mags with ONT. Eukaryote mags are rare because genomes are usually too complex to assemble and bin in metagenomes. That said, we were able to recover complete genomes for these two taxa of green algae, Bathycoccus and Ostriococcus clade B. These are particularly interesting to us because Ostriococcus is the second most abundant microorganism in the Great Barrier Reef. And because we have these T2T assemblies, we could start to see structural variation between our reef lineages and some from the Atlantic Ocean. For example, our Ostriococcus from the Great Barrier Reef is about 99% similar by ANI to the one recovered from the Atlantic, but our version has a big 300 kb translocation from chromosome 8 to chromosome 12, which we can only see because we have full chromosome assemblies. So without that, this would kind of like mask that variation. The same sort of structural variation occurs in Bathycoccus as well, where we have two variants, and one shows a 350 kb inversion on chromosome 4. And we wouldn't be able to see that without nanopore. The recovery of eukaryote mags is pretty rare at this point, but we're hoping that these results encourage more people to look into sequencing of uncultured taxa. And the last point I wanted to make is that this database is fairly unique in recovering prokaryotes, viruses, plasmids, and eukaryotes. Most databases focus on prokaryotes and they recruit between 5 and 40% of metagenomic reads. And the biggest collection of hundreds of studies, that database recruits about 40 to 60%. Combining all of our data from the prokaryotes, viruses, and eukaryotes, we recruit about 70%, which means that we can finally start to look at more of the full diversity of microbial life from the GBR. And with that, I'd like to say thank you to my collaborators from IMOST, CSIRO, AIMS, and UQ. This work is the work of a whole team, so I just have to say thank you there.